So let's talk a little more about DNA. So DNA, it consists of two complementary strands in opposite directions. That is, one is from 5 prime to 3 prime, the other will be 3 prime to 5 prime. It consists of various nucleotides. There are purines or pyrimidines. The purines are adenine or guanine, and pyrimidines are thymine or cytosine. The number of purines, the total number of purines will always be equal to the total number of pyrimidines. This is the Chargas rule. It may be asked in Krok. So DNA replicates by a semi-conservative method, that is, one new strand is found that is complementary to the parental strand. Quickly revising the enzymes of DNA replication, helicase unwinds the DNA. I'll show you in this picture. Helicase unzips the DNA. There are two strands, they are unzipping. And then there is topoisomerase that removes the negative coils, positive and negative coils. So if two strands are, it is unzipping, the coils at the end will tighten and tighten and tighten. So to remove those coils, the topoisomerase gets into action. And then we have DNA polymerase that obviously synthesizes the new DNA chain in 5 prime to 3 prime direction. Next, we have DNA ligase links the short DNA, that is Okazaki fragments. So, since the DNA polymerase only works in 5 prime to 3 prime, so if the DNA unzipping in this direction, as the DNA polymerase can, if helicase is unzipping in this direction, the DNA polymerase can continuously make this strand without any interruption. However, it is not possible for the lagging strand, because if first DNA was unzipped up to here, first it will replicate and it will make a new strand in this direction, but it will unzip more, so it will again have to work in this direction. The DNA will unzip more and it will again start making a new DNA strands. So in this way, small DNA strands are formed that are called the Okazaki fragments. In the end, since we have to make a continuous DNA strand, these Okazaki fragments are joined with the help of DNA ligase. Another thing, so DNA, if it replicates itself, obviously it is called replication. The enzyme that takes place is, the main enzyme is the DNA polymerase. From DNA, we can make RNA through the process called transcription, and the main enzyme is RNA polymerase. And the process from which proteins are made from RNA, it is called translation. So we have already talked about replication. Let's go on to transcription. It is mainly related to biochemistry, but what part is related to medical biology? Let's discuss that. And that is the lag operon. So lag operon, the genes in the operon encode for proteins that allow E. coli to use lactose as an energy source. So normally E. coli utilizes glucose, but if glucose is absent and lactose is present, so obviously it needs some enzymes to digest those lactose. So how are these enzymes produced? Enzymes are basically proteins, and for proteins there should be transcription and translation. So let's see what lag operon consists of. So obviously for lag operon to take place, you need to have a lactose. It will only occur if lactose is present. So first of all, you should know that there is a repressor. Repressor means what? It represses, it stops, or it doesn't allow a process to take place. So obviously if it binds to the operator, it blocks the RNA polymerase from binding to promoter. You'll understand. And so the transcription does not take place. The gene producing repressor is called regulator gene. Promoter. So promoter is the binding site of RNA polymerase, and if RNA polymerase, obviously its function is transcription, the start of transcription, and operator is the binding site of repressor. Terminator, obviously name itself tells, ends the transcription. So normally when lactose is absent, so obviously when lactose is absent, why is there need to produce an enzyme? So obviously transcription will not take place, and to block the transcription, the repressor binds with operator. See in this picture, repressor, if it is bound to operator, it will not allow the RNA polymerase to work and transcription will not take place. However, if lactose is present, lactose will obviously take a repressor with it, so RNA polymerase, so operator will be free and RNA polymerase can start the transcription. So again, the repressor binds to operator and blocks, and when it is bound to operator, it blocks RNA polymerase from working. And when lactose is present, it acts as inducer. So obviously when lactose is present, we need enzymes. So transcription must take place. And how transcription will take place? If the repressor is stopped from blocking operator, which in turn unblocks the RNA polymerase. So obviously when lactose is present, it binds to repressor. So repressor can no longer block the operator or RNA polymerase and transcription occurs and the enzymes can be produced. So now from DNA, RNAs, mRNA is produced, but this mRNA is still have some, it is not fully mature yet. And to make a mature RNA from the immature one, the process is called mRNA processing or RNA splicing. So obviously precursor is transformed to the mature mRNA. So there are some regions in RNA that do not code for anything, but they are important some way or the other that you don't need to know now. And obviously then there are exons that are coding regions of RNA. So first of all, what happens in this process that a 7-methyl guanosine cap is added to the 5' end of this transcript. This is basically to prevent it from digestion or to anything that can break down this newly formed transcript. Then obviously a polyadenine tail is added to the 3' end. And now when this RNA is safe from the digestion of other enzymes, now the introns can be removed. Why need to remove? Because they're non-coding, they're not useful. So obviously better to remove them when translating. So the remaining exons, they are joined by DNA ligases. That is as simple, and that is all you need to know about mRNA processing or RNA splicing. So you should know that introns are mainly present in eukaryotes. So obviously this whole process is taking place in eukaryotes and not prokaryotes. Now the transcription is done, mRNA is formed, the precursor is now turned into a mature mRNA, and now it is the time to form proteins. And proteins, as you know, are made from amino acids. So for the aspect of translation, I'm going to discuss codons. So codons are trinucleotide sequence of DNA or RNA that correspond to a specific amino acid. So here you can see there are all in the 3. So they are composed of 3 nucleotides. And these 3 nucleotides, they have code for a specific amino acid. So there are start codon, that is the first codon in mRNA that undergoes translation, and it is always AUG. Here you can see AUG, mRNA, it is in the very start of codon and it codes for methionine. Next, we have stop codons, and it is obviously for termination of translation. It can be UA, AUG, and UAG, and they do not code for any amino acid. Now let's look at this question. In the process of their translation, The first amino acid of these proteins is the same. Why? Because the start codon AUG, it is the first codon every mRNA has. So an AUG always codes for methionine. So obviously every protein, the proteins, the first amino acid will always be the same, that is methionine. Now, since we are talking about codon, let's also talk about the properties of this genetic code. First, triplet nature. We already have discussed that it consists of 3 nucleotides. Next, collinearity, comma-less. You can see in this picture, AUG, after AUG there is no comma or no dot, so they are collinear. After one codon there is another codon, then another codon. There is nothing else in between. So they are collinear. Next, degeneracy. One amino acid is coded by many codons. So you can see this table. We have first letter, second letter, and third letter because codon is triplet. You can see methionine and tryptophan, they are coded only by one codon. But when it comes to other amino acids, for example, valine, it is coded by so many codons. So this property that one amino acid is coded by many codons, it is called degeneracy. Now I also want you to notice that, for example, you can see GUU, GUC, GUA, GUG. What is the difference? What is the only difference between all these codons? That is, the third letter is different. Only the third letter is different, and still all they code for same amino acid, that is valine. I told you that, we'll see in the question. Next property, that is universality. So codons are same for all organisms. If I have AUG, then probably other animal also has AUG. Or if I have UA or UAG, UG or any other codons, other animals also have the same set of codons. This is universality. The next point, do not confuse with the universality, is unambiguous. That is, one codon will always code for the same amino acid. If in my body AUG is coding for methionine, in other animals also AUG is coding for methionine. This is unambiguous property, or in Krok it is often referred to as specificity. That is, specificity you can obviously remember, like one specific codon will code for a specific amino acid. Let's read some questions. These are questions from the ministry book. Question number 42. Under the influence of mutation in a gene, the composition of some triplets was changed. But the cell continued the synthesis of the same protein. Now why? See, they're saying some mutation happened and the cell was still synthesizing the same amino acid. How is it possible? For example, if mutation happened and from GUU it changed from GUA, it is still synthesizing the same amino acid. And this property, obviously, it is degeneracy, because one amino acid is coded by many codons. So even if a mutation occurs, the codon changes, it can still synthesize the same amino acid. Next question. It was determined that mRNA tripletic sequence totally correspond to the amino acid sequence in the polypeptide chain. What do we call this genetic code? Collinearity. If there are 3 codons, 3 amino acids will be there. If there are 5 codons, 5 amino acids will be formed in the polypeptide chain, because they are continuous. One codon after another codon, then another codon. There is nothing in between. So this property is collinearity. Next, treatment of a patient involved gene therapy. The enzyme gene was introduced into patient cell by means of retrovirus. So what property of genetic code allows to use retroviruses as a special vector of functional genes? So universality, obviously, because I told you if I have some set of genes, same set of codons are other animal, or even in this case are talking about virus, they also have the same set of codons. So what property is this? This is universality, or the genetic code is universal, same in all. So last question, it is known that genetic code is degenerate and has triplet nature. What nucleotide can be changed in the codifying triplet without losing its sense? Without losing its sense means it will code for the same amino acid. And how is it possible? I showed you in this picture. You can see here, 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 or here. Only the third nucleotide is different. First and second are same, and they are still coding for the same amino acid. So here the answer will be third. Let's quickly also talk about DNA repair. It's just a process in which the cell identifies and corrects the damage to the DNA molecule. So within the cell cycle also there are various checkpoints for the DNA, that is G1 to S or G2M boundaries. Also an intra-S checkpoint, that is the phase in which the DNA doubles, and in during the replication if they want to, the cell wants to check that it hasn't made any mistakes, so there are various checkpoints. And the enzyme, for Krok purposes you should just know the name of this enzyme, that is the endonuclease. Endonuclease is responsible for DNA repair. And also there are many other disorders. This topic also is mainly related to biochemistry, but 1-2 questions are asked in medical biology, so I have only written that much information that is asked in medical biology. So next, so in the cells of a person who has worked in Chernobyl exclusion zone, so obviously there are many radiations can lead to many mutations in the DNA. So however, over course of time the original DNA structure has been restored with specific enzymes. Obviously DNA repair has occurred. And what enzyme? If they had asked which enzyme was responsible for the restoration of original DNA structure, that will be endonuclease. Next, the most common Xeroderma pigmentosum, and they have already mentioned that hereditary deficiency of endonuclease, and the process disturbed is DNA repair. So that's it for this video. Please stay tuned for more keywords related to medical biology. Thank you.